Well, great. Thank you everyone for attending my talk. I'm excited to be here, and I'm going to present research where I investigated the evolutionary potential of the tallgrass prairie annual plant, Chamaecrista fasciculata, to changing climates in Minnesota. So this study took place in the tallgrass prairie region of Minnesota. And on this map of Minnesota here, the yellow shows the historic extent of native prairie before European colonization. And the red, which is hard to see, shows what remains, and that is about less than 1%. And additionally, the remaining prairie exists as small and really fragmented patches. So because of this fragmentation, as the climate changes, it's unlikely that prairie plant communities will be able to migrate into more favorable habitat. So the persistence of these populations may depend on their capacity to adapt in their current patches. The genetic diversity of these populations, however, might be low and insufficient for adaptation, as they are likely severed from gene flow and as a result might be subjected to processes like drift and inbreeding depression that can reduce genetic diversity and fitness and therefore population persistence. So the motivation for this study was really to evaluate if plant populations in these tallgrass prairie patches have the capacity to adapt to climate change. And by adapt, I mean an increase in population mean fitness through genetic change. So to assess whether or not populations possess the capacity to adapt to climate change, or if they have, assessing what their adaptive capacity is, we can use theory developed by Ronald Fisher at the beginning of the 20th century. So Fisher developed his fundamental theorem of natural selection, which states that the rate of adaptation of a population, so the change in mean fitness between subsequent generations, that is equal to the ratio of the population's additive genetic variance for fitness to its mean fitness. So additive genetic variance, represented by V sub A, that's the portion of genetic variance of a quantitative trait, so traits that have to be measured, like fitness, that is available for selection to act on. So fitness here, represented by W, is defined as the number of offspring that an individual contributes to the population over its lifetime. So when we're working with plants, that's the number of seeds. And then the change in mean fitness between generations, shown here as delta W on the figure, that's the rate of adaptation. So it follows that mean fitness of the progeny generation can be predicted from this ratio of the additive genetic variance for fitness to mean fitness, plus the mean fitness of the parental generation. So while we now have some evidence for the immediate potential for ongoing adaptation for wild populations of Chamaecrista fasciculata, what we still don't know is if populations possess sufficient additive genetic variation for adaptation to environments predicted for the future. Nor do we know how well predictions of the rate of adaptation from the FTNS match what is realized in the field. So to gain a better understanding of the rate at which populations can increase their mean fitness in environments predicted for the future, we used one wild population of Chamaecrista fasciculata from the prairie region in Minnesota and exposed it to novel climatic conditions. So getting into some specific research questions, to determine the adaptive capacity of our focal population of Chamaecrista fasciculata to climate change, we first assessed whether or not it had detectable additive genetic variance for fitness when it was grown in these environments representative of its future climate. And then we also evaluated if its adaptive capacity was sufficient enough to maintain or increase population growth in the subsequent generation. And then lastly, we compared predictions of the rate of adaptation from the FTNS to what was realized in the field by the progeny generation. And we did this in order to see how well the fundamental theorem of natural selection predicts the rate of adaptation. We used a space for time substitution field design with 3 sites shown here. So ML in eastern Minnesota, which was the home site, LB in western Minnesota is the middle site, and then SD is in South Dakota, and that's the westernmost site. And these 3 sites were on an aridity gradient representing climate change. So the sites get warmer and drier east to west, so away from the ML site. I conducted hand crosses in the greenhouse with seeds sourced from the home site, the ML site, and I produced a paternal half-sibling pedigreed seed set, which was then sown back into each of these sites. So estimates of the adaptive capacity of this McCarthy Lake, eastern Minnesota population planted into these western sites will give us insight into the capacity of this population to adapt to climate change. And that's because the climate conditions from these western sites are predicted to impinge upon the home site in the future. At each of these sites, I planted into about 3 acres of restored prairie, and I randomized and marked 30 seeds from each plant family. I had about 150 plant families into marked 50-meter rows. And then for each seed planted, I collected life history data, which I then used to estimate the additive genetic variance for fitness and mean lifetime fitness of the population at each of these sites. So pedigreed seed from the parental generation, so again, that was produced in the greenhouse, was sown into each site for 2 years. Additionally, in the second year, I also sowed progeny from the parental generation that was planted in the first year. So from the parental generation in year 1, I estimated the rate of adaptation, and I predicted mean fitness of the progeny generation using the FTNS. And then in the second year, planting the parental and progeny generation contemporaneously, so in the same environment at the same time, that enabled an estimation of the realized rate of adaptation. And that is the genetic change in mean fitness. So it's a change in mean fitness between generations that's due to a change in allele frequency from natural selection that happened in the parental generation in the first year. And again, this is estimated as the difference in mean fitness between the progeny and parental generation in the second year. So because these generations experience the same environment when they're planted in the same year at the same site, this method enables you to isolate the difference in mean fitness due to genes, not environment. Okay, so I am going to show you a series of results using this figure. So there are 3 panels, 1 for each site. They're ordered east to west. So McCarthy Lake is the home site, ML, and then LB is the middle site again, and SD is the westernmost site. And the LB and SD sites again represent future climate of the home site, ML. Mean lifetime fitness is on the y-axis, and you can notice the difference in scales between sites. The dashed line here is at a mean fitness of 1. And that indicates where the population would be if it was at replacement. So fitness below this line indicates population decline, and then above this line is population growth. So this first panel shows mean fitness of the parental generation at each site in the first year. And you can see that mean fitness of this parental generation in year 1 declined away from the home site. So it's greatest in the home site. However, while it fell below replacement in the middle site, it remained above replacement at SD, the westernmost site. So this shows that fitness does decrease with distance, but in this case, it's not linear. And importantly, fitness, while it declined, it's still above replacement at the western site, which means that the population here is still growing. It's not declining. All right, so in this slide, we're comparing the prediction of progeny fitness from the FTNS with what was realized in the field. So the second point for each site is the prediction of progeny fitness from the FTNS. And the fourth point in each site, so the last point, that's the estimate of progeny generation mean fitness that we took from the field. So we can see that at each site, the FTNS predicted a pretty large increase in progeny generation mean fitness from the parental generation, whereas actual progeny generation mean fitness is much lower, and in some cases lower than the parental generation. However, we get a different story when we compare the genetic change in mean fitness. So this is the realized rate of adaptation, and again, this is the difference between the last 2 points in each site. So this genetic change in mean fitness is the difference between the parental and progeny generation in the second year, and it gives an estimate of the realized rate of adaptation. So it is negative at this middle site, which indicates that there was a decline in mean fitness in a genetic sense. So there was maladaptation. But it's positive at the westernmost site, South Dakota, the SD site. And this means that selection on the parental generation in year 1, so that first point, selection on that generation, resulted in adaptation of the progeny generation to the environment that they are now experiencing in the second year. So this positive rate of adaptation suggests that this population of Chamaecrista fasciculata, sourced from the home site, may possess the capacity to adapt to climates predicted for the future, without the introduction of seed from other sites whose current climate, might be similar to what to expect for the future. So you might be wondering, so why are predictions of the rate of adaptation so different from the FTNS, so different from the actual rate of adaptation? So importantly, the fundamental theorem of natural selection assumes that the environment of the progeny will be the same as its preceding generation. And we just know that this is not true because of interannual environmental change and long-term climate change. So we don't expect predictions of mean fitness from the FTNS to be a perfect match of what is realized in the field. But the accuracy of the FTNS has never been evaluated, so we don't know the extent to which predictions will deviate. To kind of untangle what might be going on here, we can estimate breeding values of plant families that I had from the pedigree to kind of gain insight into how the selective environment may have changed between years. So breeding values are the effect of an individual's genes on trait expression of its offspring, and that's due to additive effects. And this panel here shows 3 different figures, each for each site, and they show breeding values of the parental generation for each site compared between both years. So the parental generation in year 1 compared to the parental generation in year 2. And I calculated correlation. I calculated the correlation between these breeding values for each site, and I found that they were weakly or to moderately correlated. And this means that families with high fitness in 1 year are poor predictors of their fitness in the second year, which suggests that the selective environment changed between years and may explain the poor prediction from the FTNS. So what do these results mean for the adaptive capacity of this population of Chamaecrista fasciculata to future climates? First, the detection of substantial and significant additive genetic variants for fitness of the parental generation in both years at all sites suggests that this population does possess the capacity to adapt to environments predicted for the future. There's tons of heritable additive variation for selection to act on. And this additive genetic variance for fitness that we detected, along with mean fitness of the first parental generation, it was abundant enough that mean fitness of the next generation, the progeny generation, was predicted to be above replacement in future climates, indicating population growth. But the prediction did not match what was observed in the field, likely due to interannual environmental change. But importantly, mean fitness of the progeny generation at the westernmost site, representing, a potential climate future of the home site, remained above replacement, and it had a positive rate of adaptation. And this suggests that the capacity of this home population is present to adapt to future climate. So while I found that a change in the selective environment between years can really reduce the accuracy of the prediction of adaptation from the fundamental theorem of natural selection, it's still an important conceptual tool, and hopefully with more studies, we can refine predictions and gain a better understanding of the rate at which populations can increase their mean fitness in response to climate change. So I'll just do a quick thank you to all my funding sources and people who helped make this research possible. And I just wanted to bring your attention to the work I'm currently doing briefly with the Dr. Ruth Shaw Research Group at the University of Minnesota Twin Cities. I'm a postdoc for the Healthy Prairies Project, as was mentioned in the introduction. We're in our 8th year of this project, I think 8th and final year, and we have 3 aims in which to help understand and promote prairie conservation. So we are doing seed collections, we're evaluating plant microbe associations, and the third arm that I'm most involved with is looking at the geographic scale of adaptation. So we have 6 perennial tallgrass prairie plant species in Minnesota that we've planted into 3 common gardens, and we're currently evaluating and writing up work about the scale of adaptation for these species, with the hopes to help inform seed sourcing decisions for land managers. So I'm happy to talk to anyone if you're interested in that, and you can also take a look at our website if you are curious for more information. And that's all I have, and I am happy to take questions.